This webinar will also be posted on the Facebook page of the Philippine Planktology. So if you have friends who haven't registered for this Zoom webinar, you can tell them to go straight to the FB page of Philippine Planktology. Thank you. Good afternoon, everyone. Welcome to the second Philippine Planktology Lecture Series. We have participants today coming from all over the Philippines, as usual. Thank you again for waiting patiently in the waiting room. And then also for those watching on Facebook Live, welcome to this webinar. So this is, today we will have our last, but of course not the least, speaker or lecturer, Dr. Shuhei Ban. So previously we had a lecture from Dr. Yuji Machida and Dr. Chihao She. So thank you very much for our speakers. Okay, so just a few reminders of the house rules. For the participants, we are requesting everyone to turn off your cameras, and then please keep your microphones on mute. And please hold your questions until the end of the presentation. And these questions will be entertained later during the open forum with Dr. Louis Lopez as our moderator. To ask a question during the open forum, you can raise your hand if you want to ask a question live. Or you may also type in your questions in the Q&A box, and also on the FB live stream. We will have people working together to gather all these questions so we can have them entertained by our lecturer later. And then we will post the evaluation form later on our Facebook page, the Philippine Planktology, for you to get your electronic certificate. Okay, so I think we are right on time. I believe that we had a little bit of misunderstanding. The program starts at 2 p.m. And then thank you again for those who waited in the waiting room. And then we will have our director to introduce the speaker, and then the talk, and then later the open forum and closing remarks. And hopefully we can have a photo taken of the group. Okay, so right now I would like to welcome the director of the Institute of Marine Fisheries and Oceanology, College of Fisheries and Ocean Sciences, Dr. Sheila May Santander de Leon, to give the opening remarks.
Thank you, Christy. Good afternoon, everyone. Welcome to Historia Lawood, Stories of Our Oceans. We have conducted, I guess, more than 8 sessions already just from July this year. This is because we have so much information to share with you together with our research partners and stakeholders. This afternoon will be another story on planktology, which is the last of the 3 installations. I'd like to share this opportunity to welcome and thank you, Dr. Shuhei Ban. Watashi tachi wa sensei no hanashi wo kiki anata no kenkyu kara manabu koto wo netsubu shitemasu. Honto ni arigatou gozaimashita, sensei. Again, welcome everyone.
Thank you very much, Director Sheila. Okay, so now let us proceed. Okay, I'd like to introduce our lecturer for today. He is a professor at the Department of Ecosystem Studies, University of Shiga Prefecture. Dr. Ban's expertise include plankton ecology and limnology. His works mainly focus on the physiological effects of temperature, food availability, and predation on the growth and reproduction of freshwater zooplankton in Lake Biwa, Japan. So if you remember previously, our speaker, Dr. Zach, also talked about Lake Biwa. So this is a very good transition to our third speaker. Please welcome Dr. Shuhei Ban.
Hello, everyone. I'm Shuhei Ban from University of Shiga Prefecture, Japan. Nice to see you. And thank you for inviting me to this interesting lecture series, Louis and Christine. Our university is located in Hikone of Shiga Prefecture and just next to Lake Biwa, the biggest lake in Japan. Shiga Prefecture is located next to Kyoto and the central part of Japanese mainland, Honshu. Hikone is countryside and having a lot of beautiful sceneries. If you will have a chance to come to Japan, please visit our city and our university. Anyway, I'm now studying every kind of subject in freshwater environment. From nutrient cycling in the lake to laboratory study on zooplankton and mass culturing microalgae to treat wastewater. But I'm fundamentally zooplankton ecologist. Today, I'd like to talk about effect of temperature and food conditions on zooplankton growth and biomass. Although you know a lot of environmental variables that potentially affect zooplankton life history traits, these 2 environmental factors are the most influential variables for understanding zooplankton physiological activity. And additionally, I'd like to talk about top-down control. Top-down control such as predation often mask these 2 effects. Can I share my presentation file?
Yes, Sensei, you can now share.
Can you see the slide?
Yes, we can.
Okay. These are today's topics. Firstly, I will introduce the effect of temperature on zooplankton physiological rates because zooplankton are mostly placosomes. So one of the most important variables in temperature is temperature. Next, I will talk about effects of food abundance and quality on growth rate. Finally, I'd like to explain the effect of predation by fish as one of the biological and environmental variables. Such top-down effects often influence zooplankton abundance, distribution, and behavior. and may consequently mediate results of physiological effects. So I will start from temperature effect. As I already mentioned, zooplankton are mostly poikilosome, meaning their physiological rate depend on surrounding temperature, because most biological processes, including enzyme activities, are temperature function. Therefore, temperature is one of the most influential parameters for zooplankton growth and reproduction, and consequently, its abundance and distribution. These are the temperature-mediated physiological variables in zooplankton. Embryonic development time means egg development time, or the time from laying eggs to hatching. Post-embryonic development time means juvenile development time, the time from hatching to adulthood. Growth rate means increment rate of body weight, and feeding rate, respiration rate, excretion rate, and so on. One of the representative is an embryonic development time that might be affected mostly by temperature, but not food, because the developmental processes proceed with reserved egg yolk and same size of the eggs that produced even by the different species. Actually, egg size also influences the development time, and it has been shown that larger eggs need longer development times. Indefinite egg size increases with increasing the female body size and age. So embryonic development time may also increase with female size and age. Anyway, generally, the embryonic development time has been shown to exponentially increase with decreasing temperature, like this, the figures. A lot of the model for temperature function has been proposed as described in this slide. The Bell-like function, hyperbolic function, power function, exponential function, and its relatives. One of the most famous and recommended model is Bell-like function, this one. A reason why this model is chosen is that one of the constant of alpha is included. This constant is considered to be indicated as a threshold of lower limit of physiological activity, so it's called biological zero. For this example and result of erythemal affinity that I studied when I was graduate student, alpha is here, minus 3.4. This figure shows the relationship between alpha as biological zero and average environmental temperature among the different species of copepods colonizing in different temperature environment. McLaren 1969 find that when Bell-like functions B is fixed at minus 2.05, the value of alpha linearly increased with increasing mean environmental temperature experienced by the copepods. By the way, as I mentioned above, embryonic development time is a convenient parameter for understanding a physiological adaptation of the animals to the surrounding temperature. And it might also be one of the easily but actually measured parameter. If you wish to measure the embryonic development time precisely, how do you have to do that? Do you measure the time from laying egg to hatching nucleus? Please try to imagine the scene that you would be doing such kind of experiment. It seems a quite tedious work, but not necessarily to be measured precisely. One of the convenient and precise measurement protocols has been proposed by Edmondson in 1965, more than 50 years ago. I'd like to introduce this nice method briefly. If a natural population of your target copepod, Daphnes or rotifers, would randomly or non-synchronously produce eggs, the eggs collected at the same time should be hatched at various times within the real development time. So you collect a lot of egg-carrying female from the field, detach them, and incubate them at an appropriate temperature. Then you check the hatching the eggs in an appropriate interval. And finally, you can plot a proportion of unhatched eggs at each of the observation time from the start of the incubation. This kind of, you can get this kind of the figures. Then you can get the result like these figures. This is one of the example for that kind of experiment. We measured embryonic development time for Eodiatum japonicus collected from Lake Biwa. As you can see, after the regression analysis, the time at 0% of unhatched eggs can be precisely calculated as mean development time of in situ population that you collected. Okay, let's move to the next, post-embryonic development. Next is the post-embryonic development, which means juvenile development from hatching to the adulthood. Therefore, it does not only influence temperature, but also food condition. Actually, until more than 40 years ago, a lot of biological oceanographers believed that the copepod growth and development depended on temperature alone, and food effects are generally negligible in nature. This is the most influential paper of such temperature-dependent hypotheses. Hunter and Lopez, 1992, showed that temperature is the most predictable variable for copepod generation time and specific growth rate irrespective of food condition. They collected dataset from the previous literature, including 33 copepod species and various regions of ocean with various food conditions. This graph showed that the generation time, that is the period from hatching to adult, they're almost the same as post-embryonic development time. It were well explained by temperature alone with this exponential equation. It was quite important finding because generation times for any copepod species from any ocean regions can be calculated from this equation and temperature equation and temperature alone. However, you should be careful to see that they have a large variance here. In turn, these are the specific growth rate, which are calculated from dry weight of egg and adult, and generation time from the literatures. In this the equation, WA is adult weight, WE is egg weight, and tau is the generation time. These are also strongly depend on temperature, but showing large variance yet, especially at the high temperature. Needless to say that these variances might be attributed to environmental variables other than temperature, probably food conditions in most cases. Actually, in 1984, International Symposium for Food Limitation on Zooplankton Community has been held in Germany. So Huntley and Lopez 1992 was a challenging paper for the concept of the importance of food limitation. They believed that marine carpetboats were not suffered with food limitation as previously believed in biological oceanography. But I think this was not true even in marine environment. Okay, move to next, food effect. Food conditions are divided by the two different components, that is, food quantity and food quality. As you know well, food abundance is an important factor for regulating growth, development, and egg production. That is, cell number or carbon biomass of food algae per unit volume for feeding zooplankton. On the other hand, how does the food quality influence those life history traits? Food quality is, for example, represented as elemental ratio, C to N2P ratio, lipid contents, for example, the polyunsaturated fatty acid, and some toxic substances, and so on. This is one of the examples. This is the experimental result for understanding synergistic effect of food algal concentration and temperature on freshwater calmated carpetbot. Y-axis represent body dry weight of the carpetbot, and x-axis represent days from hatching to days from hatching. So these show the growth curve. Upper panels show the results at 15 degrees C. Lower panels show those at the 20 degrees C. M represent male, F is female. Please see the two bottom panels first. The food concentration decreased from left to the right. As you can see, the growth is the highest at the highest food concentration and decrease with decreasing food concentration at both male and female. Interesting thing is that this food depletion equation is not the same as those found in lower temperature. The food effect is not so much at lower temperature. To make easier understand this synergistic effect of temperature and food concentration, you can calculate food growth coefficient with an appropriate function from these curves. For example, that is Bertrand-Pick's function. You can fit your result with this function, for example, with a nonlinear least square method, and calculate the growth coefficient. Here is a K. These are the results. As you can see, this modification seems to be better understand the synergistic effect of food and temperature. Y-axis is growth coefficient and x-axis is log transformed food concentration. According to the results, the growth rate of the carpetbot increased with increasing food concentration at 25 degrees C, but almost the same at the 15 degrees C. And consequently, the growth rate at 25 degrees C were more than double of that at the 15 degrees C at the highest food concentration. But this difference reduced with decreasing, food concentration. And finally, almost the same at the lowest food concentration. This interaction effect between temperature and food concentration might be attributed to lower metabolic demand or respiration loss of carbon at lower temperature. On the other words, lower food uptake, high respiration loss at high temperature depresses the growth, but not so much depression at lower temperature. This also means that food effect might be severe in warm seasons, but not winter. In winter, generally food abundance is not so much because phytoplankton production depresses, but probably low temperature is dominant reason for lowering the growth. Next is food quality. This slide shows experiment for effect of food quantity and quality on definite growth. Y-axis represent body dry weight of the experimental animals, and x-axis represent days from hatching. So these curves directly show the growth of the animals. Vertical column means food concentration. Left one is the lowest food concentration as dry weight of the animals, dry weight of the food algae per liter. Middle one is middle amount, and the right one is the highest amount of food. Whereas horizontal row means the food quality. In this experiment, food algae were rarely in different nutrient supply. The sufficient food supply of nutrient in the upper panels, the nitrogen limited in the middle one, and the bottom panels in a phosphorus limited. Firstly, pay attention to upper panels at sufficient nutrient supply experiment. The highest growth is found at highest food level. This is the same as those in the capipod that I mentioned earlier. Next, you can see the right panels at the highest food concentration. The definite growth decreased when fed with nutrient-limited algal food, especially phosphorus-limited one, even at the enough food supply as dry weight. This demonstrates that definite growth cannot be explained by food quantity, but also food quality. An important thing is phosphorus content of the food items in the case of definite. This means that elemental ratio of the diet is an important for understanding zooplankton growth in nature, because this kind of food quality and elemental ratios of food particles in the field are known to be quite variable. Such kind of balance of elemental ratios between zooplankton and its food is related to so-called ecological stoichiometry. As you know, stoichiometry is a study field dealing with the relationship between quantity of reactants and its products to chemical reaction. For example, process of photosynthesis and respiration like this. Ecological stoichiometry expands this concept to prey-predator interaction to a food chain. This relationship depends on different responses to homeostasis between producers and consumers. Producers' body elemental ratio, for example, N to P ratio of algal cell, depend on nutrient elemental ratios surrounding the algal cell. While consumers' body elemental ratios, for example, P content, is relatively constant of food, irrespective of food phosphorus content. In other words, consumers have a homeostasis, but producers do not have such ability. Since consumers' elemental ratio is constant due to homeostasis through food chain, the difference of the elemental ratio affect the growth rate of the consumers. When phytoplanktons N2P ratio will be larger than that in zooplankton, zooplankton growth would be limited by phosphorus. In turn, it would be smaller. Zooplankton growth would be limited by nitrogen. In summary of the story until now, temperature is the most important factor for understanding physiological activities and life history traits of zooplankton. And food quantity is also quite important influential variable that the effect reduces in low temperature. Food quality also affects growth and reproduction of zooplankton through limitation of elemental essential element even under sufficient food supply as dry mass or carbon weight. However, these effects of temperature and food condition are often disturbed by other environmental factors such as predation, competition, and destructive physical disturbance like storms. These factors often mask the effect of temperature and food condition on spatial-temporal distribution of zooplankton. Here I introduce the effect of predation by fish on in situ zooplankton biomass and site distribution. Such strong top-down control may often exceed the effect of temperature and food condition. This is the most famous study on fish predation effect on zooplankton community in Brooks and Dodson 1965, published in Science. This slide shows zooplankton size distribution in a North American lake in 1942 and 1964. In this lake, small fish as plankton feeder. Oh, sorry. In this lake, a small fish as planktivore increased during these 20 years, and zooplankton size distribution shifted from larger than 0.7 millimeter to less than 0.4 millimeter. This destructive change of size distribution of zooplankton might be attributed to fish predation through selective feeding of the fish, because fish selectively fed larger zooplankton. This predation impact does not only influence the size distribution, but also biomass, and consequently influences their spatial temporal distribution and maybe phenology. This is an example for our own study. These are the year-to-year variation of each dominant crustacean zooplankton biomass in Lake Biwa during the last 40 years. At a glance, the year-to-year variation are different among the zooplankton species, and it implies that the factors influence these biomass variation are different among the species. If temperature is only one dominant influential variable, all species seem to respond in the same trend that actually no. These are the long-term variation of annual mean water temperature above 20 meter, total phosphorus concentration, and fish catch of Ayu in Lake Biwa during the last 40 years. Total phosphorus is mostly correlated to chlorophyll A concentration in Lake Biwa, and therefore it can be used as a food proxy for zooplankton. Ayu is a typical planktivorous fish in the lake and one of the dominant planktivora in Lake Biwa. CPUE is an abbreviation of catch per unit effort and can be used as a proxy of predation impact by planktivorous fish. Lake Biwa experienced a regime shift during 1980s, and water temperature increased approximately 1 degree C in mixing layer of the lake after 1985. Total phosphorus concentration increased until 1970 due to eutrophication, but decreased after 1980 because of regulation of point source nutrient loading by local government acts. IUCPUE increased until 1990, but decreased toward 2005, then increased again. These show annual mean body size and biomass of total crustacean zooplankton, body size and biomass in Lake Biwa during the last 40 years. And bottom is IUCPUE. Both body size and biomass decreased until 1990 and then increased thereafter, and seems to be correlated to IUCPUE. These are the results of correlation analysis between biomass and total phosphorus, biomass and temperature, and biomass, body size of zooplankton, and IUCPUE, biomass and IUCPUE. Both of the top two are not significant, but both of bottom two are significantly correlated. This means that biomass variation of crustacean zooplankton cannot be explained by both temperature and food proxy, while both body size and biomass strongly depended on fish predation. It's surprising that zooplankton biomass did not increase even in the eutrophication period of the lake. You remember the famous example, Brooks and Dodson, that I mentioned earlier. Planktivorous fish select larger zooplankton, and consequently, the size distribution was skewed to smaller zooplankton. So in this case, this negative correlation between biomass and CPUE should be explained by fish predation. As I mentioned earlier, both temperature and food condition are dominant influential factors on physiological activity of zooplankton. But the strong top-down control, as I explain now, may often mask the effect of these two main variables on spatio-temporal distribution and biomass of natural population of zooplankton. This is the end of my talk today. Thanks for listening. Do you have any question? Thank you very much.
Thank you very much, Dr. Ban. I would like to pass the virtual floor to Dr. Louis Lopez for the questions. Thank you.
Thank you so much for the presentation, Ban Sensei.
Thank you.
So what we did is we collected some questions from the audience. for both from Facebook and then from the chat in Zoom. And then I think I'll start with some of the basic questions coming from the students. So you mentioned calculating generation time of zooplankton species. So one of the basic questions is that uh, why do we need to calculate generation time, and what's the possible importance of this data in fisheries?
Mm, you wanna measure the generation time. You have to check the animals' development for when you use the lower temperature, and the 1 day is okay, but more higher temperature, you have to check the more shorter interval of time. For example, twice or the more than times in a day.
I see.
It's a very tedious work.
Okay. I can imagine. How about you in one of your slide, you presented the equation from Huntley and Lopez. But
Okay.
yeah. And then I think one of the audience is
This one.
asking,
Yeah.
can this also be applied to freshwater species? Because I think
No,
most
this
of
is the
them are.
marine copepods.
I see.
It's the marine copepods.
Like, do we have counterpart for this one when it comes to freshwater species?
I'm not sure recently, but maybe no one
I see.
make this kind of one, because in freshwater the food effect is very severe.
I see. Yeah.
So yeah.
And then another question from the audience. Why food effect is less significant at lower temperature? So, and then at which temperature can we start observing this pattern? I think in one of your slides you mentioned food effect is weaker at lower temperature. And then can we predict at which particular temperature can we observe this weakening effect?
So food effect usually intense at the higher temperature, because in a higher temperature the metabolic rate is high, and zooplankton. So in a high metabolic condition, they loss of energy due to the respiration. So the food effect is generally the higher temperature, but not so much in lower temperature.
Yeah. And then I think the next question is somehow connected to the previous one. It has something to do with the metabolic rate, right? And then the question is what's the best way to measure metabolic rate in zooplankton species? I think this is coming from a researcher from the audience. Like they want to know is there any method how we can measure metabolic rate in zooplankton because they are so small?
Maybe when you wanna measure the metabolic rate, the most easily measured one is the respiration rate, oxygen consumption rate. Another one is the excretion rate, ingestion rate. So if you know in a respiration rate experiment, you have only the skill of the measuring the dissolved oxygen content, measuring the dissolved
Yes.
oxygen content, and
Yeah.
for example, using the titration method, it's very
Mhm.
easy to measure the oxygen concentration in the water.
Yeah. Mhm. Yeah. And I think to add that, Sensei, I remember from your previous studies, you're using optical sensors, right? For
Yeah,
measuring.
it's a very the good one, but a
Mhm,
little
I
bit
see.
expensive.
Yeah. But is it more accurate, like in terms of the measurement overall?
Pardon?
Using Is it more accurate using the optical sensors? I think you
Yeah,
just
yeah,
attach it. You just
quite
attach it
accurate,
outside,
quite accurate. Yeah,
right? Mm-hmm.
I recommend it, but little bit expensive.
Expensive. And then I think another question from the audience, uh, with regards to food effect,
Yeah.
are we going to observe the same pattern for the food effect if the zooplankton species is actively migrating, like during the vertical diel migration? Like,
Uh-huh.
yeah, how can the vertical diel or the diel vertical migration affect food effect in zooplankton species?
Mm. And usual the zooplankton that uh, the having a deal vertical migration, and they can eat during the daytime, and they cannot eat much more cannot eat much food during the daytime because they go down to the lower temperature and the low food concentration in deep part of the lake or the ocean.
I see. Oh, yeah. And then another question is, I think needs more clarification. Comparison between phosphorus and nitrogen limitation.
Mhm.
Which one is more important in affecting body growth? Is it the nitrogen or the phosphorus limitation?
That is depending on the species.
Mhm.
And definitely the phosphorus is the important, while the copper and nitrogen and is limitation is important.
I see.
So
Since it,
depending on the demand of the essential element.
I see.
Yeah,
Is this
copper
is this also
need more nitrogen and definitely need more phosphorus.
okay? So basically, this is related to food preference of zooplankton species. Is that right? No.
Maybe どうかな。Maybe related to the food preference to their the body chemical composition.
I see.
Usually the definite is the phosphorus rich body, and
Mhm.
the copper is the nitrogen rich body.
I see.
Maybe depending on the food.
Yeah.
I'm not sure precisely.
Mhm.
Mhm.
And then another question that we have from the audience, I think, it's still related with nitrogen limitation and phosphorus limitation. Like, are we going to observe the same pattern if uh there is, like, for example, a harmful algal bloom? Because now you need to consider presence of toxins in the food availability because of the cyanobacteria present.
Mm. Um. Uh. Now, generally, the cyanobacteria is not so good food for zooplankton
Mhm.
because
Yeah,
the polyunsaturated fatty acid is not included in the cyanobacteria.
mhm. I see.
And maybe the often include the toxic substances and cyanobacteria for zooplankton too.
Yeah.
So
I see.
apart from the elemental ratios, they have the toxic and non nutritional not include the nutritional element.
Yeah. Right. And then this is a personal question. Like, I'm really amazed with the data of the zooplankton biomass that you have in Lake Biwa. I think you presented 40 years or more worth of data. Like, I'm curious which zooplankton group is more responsive in terms of the change in size structure. Is it the cladocerans or copepods?
Most dominant one is the copepod, carnoid
I see.
copepod, Eodiaptum japonicus. Next is the daphne.
I see. But in terms of changes in size structure, I think you presented a slide wherein you showed changes or disruption in size structure of the population. Yeah, that one I think. Like,
This one,
is it is it the copepod or the daphnia which is more responsive in terms of changes in size structure?
Largest zooplankton taxa is the daphne and the lake.
Mhm. Yeah.
Next is the Eodiaptum
I
japonicus.
see.
Yeah. So the most vulnerable to the fish predation is
Yeah.
daphne. Yeah.
I see. Yeah. And then, oh, okay. And then still related with the Lake Biwa, you showed that there's a disruption in temperature stability in Lake Biwa. I think it happened during 1980s, wherein
Yeah.
there is an increase in 1 degrees increase. So question from the audience is that how disruption in temperature stability affect overall zooplankton production in Lake Biwa? Does it cause increase or decrease in zooplankton productivity?
In our study, the
Mhm.
temperature does not affect the zooplankton biomass and the growth.
I see.
For example, you can see this one.
Mhm.
Temperature is not affect the zooplankton biomass. Yeah.
I see. I think we're down to last 3 questions from the audience. So another question then says it, does the regime shift in Lake Biwa impact only predator but also zooplankton composition of oh, oh yeah, I think this is already answered. Like you've mentioned that this shift in temperature doesn't really affect zooplankton production
Yeah.
overall, right? But mhm. This is maybe unrelated. Do you think it affects biomass of phytoplankton based on your data? Like, do you have phytoplankton data during this time? No.
This change?
Yeah. How about like the autotrophic or the autotrophic organisms, like the primary producers? Did you observe any change?
No change during the regime shift.
I see.
We can detect this apparent regime shift during the 1980, but any other the changes in zooplankton and the other environment, we cannot detect it as such kind of one.
I see. And then I think some of our audience are not really familiar with Lake Biwa. Some of them are asking, why did you choose studying Eudaioptimus japonicus? Like I think most of your publication would focus
Eudiotomus.
somewhat, yeah, Eudaioptimus japonicus. Is there
Yeah,
any particular reason?
we are studying the Eudiotum because the Eodioptum japonicus is the most dominant zooplankton in Lake Biwa. More than 60 to 80% of the total zooplankton biomass.
I see.
Yeah, so we are studying that zooplankton.
Okay. Is it endemic in Lake Biwa, or it's also distributed all throughout Japan? The Eudaioptimus japonicus.
Eodioptimus is the endemic species of Japan, so it's distributed all part of the Japan.
I see.
Lake Biwa is one of the habitat of them.
I see. And then I think a question that is related to that one, are we going to observe same pattern for the effect of temperature, food, and predation if your species that you're studying is an invasive species? I think one of our audience, it's mentioning Arctodioptimus dorsalis in the Philippines. It's invasive. It's also dominant and very high in abundance in most lakes. Are we going to observe same patterns, or should we expect to observe same pattern on temperature effect and food effect if you're focusing on invasive species?
So, yeah, you can observe the same kind of one as your carpetbird Arctodioptimus. Yeah.
I see.
Sure.
Yeah. I think that's all the question that we have from the audience right now. And I think if they have additional questions about your presentation or about Shiga in general, I think they can freely email you, right? Would it be okay? Like if some of the audience interested on some part that you presented, they can freely email you, Sensei. Yeah. So I'll give Christy.
Thank you very much, Dr. Ban Sensei,
you.
for your very informative talk. I believe that we have a lot of students here in the audience today, and I believe a lot more will watch from the FB Live stream later. Okay, so thank you very much. May I ask to share my screen, please? Okay. Can you please, Dr. Yes, thank you. Okay. Yes. Okay. So at this point, we would like to award a certificate to our lecturer, Dr. Shuhei Ban. Okay. So in recognition of his outstanding contribution as keynote speaker of presentation entitled "The Effects of Temperature, Food, and Predation on Zooplankton Growth and Biomass" at the second lecture series on Philippine Planktology on August 22, 2022, signed by the Director of the Institute of Marine Fisheries and Oceanology, Dr. Sheila Santander de Leon, and the Plankton Society of the Philippines, and the administrators of the Philippine Planktology. Thank you very much. Can we have a group photo for the panelists? I would like to ask Ma'am Jonaline to do this for us, please.
Thank you. Thank you very much.
Thank you for your time.
Thank you very
Ban Sensei.
much.
Hello. Ma'am, can I ask you to stop sharing, please?
Oh, yes. Okay.
Thank you. All right. And wait lang, I'll remove your spotlights so that everybody will be okay. Okay. So 1, 2, 3. Another one. 1, 2, 3. And thank you very much.
Thank you
Ban Sensei,
very much.
Thank you so
very
much for sharing your studies.
Okay. So I would like to announce to the participants, please stand by and check out our Facebook page, the Philippine Planktology page. I will post the evaluation link there later, and then we will send the certificate later on. Thank you for attending, and see you next year, I guess we will have more lecturers next year. We will be inviting other planktologists around the world and also from the Philippines, whoever is available. Thank you very much. The Institute of Marine Fisheries and Oceanology, Historia Lawood, the Plankton Society of the Philippines, and the Philippine Planktology FB page. Thank you very much. Thank you, Dr. Ban Sensei.
Thank you. Bye bye.
Hello
Thank you,
to
Dr. Ban.
Dr. Machida. And some of my friends are also here. Donna Guarte. Hello, everyone. Thank you. We may end the webinar now. Thank you.
Bye,
Thank you.
Dr. Ban.
Bye.
Bye, Dr. Ban. Bye, everyone. Thank you for your help, Sir Junior.
you.
Salamat.
Bye po. Salamat.
Bye, Louie. Good night.